Okay, can y'all see my screen? Great. So I finished grading your test last night. I have put in midterm grades. I don't know if they're showing in in Banner yet. I don't have the exact midterm grade number in Canvas that I've calculated, but you will see a letter grade in Banner when it shows up. You can see your test now. Let me check one thing real quick before I do, because I don't remember when. Okay, let me set it so that y'all can see the correct answers. Go ahead and save that. Okay, for the multiple choice, you'll be able to see the correct answers now. For the short answer, I want to go over those with y'all. And please stop me with any questions you may have. The first short answer question is who discovered the first cells, what cells were the first discovered, and what is cell theory? The first cells were, sorry, the first person to define cells was Robert Hooke. The first cells that were discovered were specifically cork cells, okay? Not just plant cells, but cork cells. And what is cell theory is that there's three parts. Cells come from other cells. Cells are the basic unit of life. And oh, phooey, now I'm forgetting the third one. And you must have at least one cell to be alive, to be, considered an organism. That's what happens when you're doing it right off the top of your head. Okay, do y'all have any questions for that one as we go along? Okay. On the next short answer question, give two things that provide evidence for endosymbiosis for mitochondria and chloroplasts, and also name, should say, well, name one other fact about mitochondria and chloroplasts that does not include evidence for endosymbiosis of the use in metabolism. Okay, the first part of this, give two things. They both have their own DNA. They can reproduce themselves. They have ribosomes and can produce their own enzymes. They have a double membrane, and they are the appropriate size to match up with either purple bacteria for mitochondria or something like cyanobacteria for chloroplasts. Now, something that's not for endosymbiosis or metabolism, what I would have chosen, although I do sometimes take other answers if I thought it was appropriate, is they are maternally inherited, and that is something that's true for both of them. So that means that the mother plant is the one that passes it on to the offspring. So the egg cell is where the mitochondria and chloroplasts are found and get passed on to the child, because the child develops in that as from that egg cell. Okay. Now, some of y'all tried to give another fact about them individually. That's not true for both, and that's not exactly what this question is looking for. Why is photorespiration a wasteful process? The biggest thing is that it's expensive. Okay, it costs more ATP to do this, and it cannot be done indefinitely. It does use oxygen instead of carbon dioxide. So it's a limited time that it can be used for that plant. Describe the light-dependent reaction. So in this case, I'm not going to go over it because I just want you to go back and refer to the diagrams that we drew out. Some of y'all just forgot some small pieces of it. Sometimes it was unclear what exactly you drew, so I couldn't give full credit if it wasn't clear. You must talk about it being a membrane-bound process, so you've got to have the membrane represented somewhere. What are the effects of light and temperature on photosynthesis? What types of photosynthesis are best under high temperatures? Light and temperature, usually there's the idea that if you increase them, you increase photosynthesis, but that's not completely true for every type of photosynthesis. Sometimes temperature will affect, because we know how temperature affects enzymes, it increases the rate of photosynthesis, but in other times it doesn't. So like with C4 plants, we see a different effect than with C3 plants. And the types of photosynthesis that are best under high temperatures are going to be things like C4 and CAM. So don't forget CAM. Light and temperature aren't always going to affect things in the same way, although they are related ideas. A lot of times having a higher amount of light means you get a lot more higher temperatures. But light can also cause problems. You can have too much light and it will actually destroy enzymes or various molecules. Temperature can do that as well, but not for exactly the same reasons. I'm not going to specifically go into the two characters for chlorophyta, cryptophyta, and phaeophyta. I think it's better if you just refer to your table. For those that did this, the answers that were written were pretty good. I think the biggest problem with this question was actually remembering something from all of these. I don't remember seeing anybody that repeated characteristics. And what I mean by that is I didn't see characteristics for chlorophyta that ended up being the same for cryptophyta or phaeophyta. All the characteristics were different. So the numbers of stacks of thylakoids, looking at flagella, and so on. Y'all were very specific when you answered it. The next one here is it's got the answer choices so that you can see those when you look at your answers. So those are there. Give 2 reasons or evidence for why the Chirophyceae and not the Chlorophyceae are the ancestors of land plants. Be careful to focus on the Chirophyceae. Now, these are both part of Chlorophyta. They're families within the phylum or domain. But Chirophyceae seems to be closer. And in this case, we have phragmoplasts, the orientation of how like the microtubules work to create the cell plate is more like plants in the Chirophyceae. We see embryophytism, or at least incipient embryophytism, which just means that it's starting to kind of hold on to its embryo for a little bit longer internally before it's released. And of course, we have a table that lists these 2 things together. So that's what I was looking for here. And this next question, this was the best picture I could find without like drawing one to compare to what we talked about in class. Why do we not use the dorsal fin for classification purposes? Because what we're seeing is convergent evolution. Mammals have a dorsal fin because they live in similar habitats to the fish. And what this means is that that feature is not useful for relationship because it's convergent. It's evolutionarily developed more than once. So dolphins are not closely related to fish. There is a relationship, but the relationship is basically every other animal is also related to fish, or at least all the other vertebrate animals. Okay, The thinking question, list 3 reasons, this was a bonus, why humans should be interested in the biology of oxygenic autotrophs. Well, they create our food, they create oxygen, they are some of the first pioneers of new habitats. They have all kinds of different products that we make from them. So anything along those, that's what I was looking for. So this is human relevance, as some of the slides are labeled. And the last one, explain why classification is important in biology. It gives example of classification and how classification is achieved in botany. So classification is important in biology because it helps us to make sure that whatever we're talking about the same thing as someone else. So scientific names are not repeated or doubled, they are specific. So when I say Rubus idaeus, I'm not talking about Rubus parviflora or Menthus bicata or I don't know, whatever you want to come up with, Fallopian japonica. I know exactly what those are when I hear those names. It also helps us to understand relationship between organisms. So we not only learn as we learn more about the plants, we can classify like what's more closely related to others than not. And how do we achieve this in botany? A lot of this is through looking at the comparisons of different organisms, different plants. So we are looking at comparisons of flowers, comparisons of vascular tissue, comparisons of all kinds of things. And so we are able to classify groups of plants based on different characteristics like morphological characteristics. We also do it now with DNA to help us understand even more closer relationships when you can't tell them either apart or tell how closely related something is through DNA. We didn't really talk about DNA, but we talked a lot about morphology. And that is exactly what we see in our tables. When we set up our tables, like with the algae, we are setting up these morphological characteristics to compare to one another. And we use that to help classify them. We group things based on these features. Do y'all have any questions? No? Okay. Let's see. Okay. No. I want to close this slide. Okay. So we've gotten all through this one, number 11. Let's go move on. If y'all don't have any questions. We've covered vascular tissues, the ground tissues. We had a pretty good little discussion for a couple of y'all that lasted on Wednesday for the last few minutes with comparisons, and I think that was useful. I think I continued the recording, so once I get those put up, you'll be able to see that. Let's look at the bryophytes. So we're about to start with the first group of land plants. And our plants of today is not a bryophyte. It's just a random other plant. This is Canophilus americanum. And it is a parasitic plant that grows, I think, on oak tree roots. Some people call it squirrel corn. I guess it looks like corn. It's got the family, the Orobanchaceae, is sometimes called the broomrape family. It's in the woods under many oaks. They only get about 3 to 8 inches tall. It's not very big. The flowers are kind of small. And by the way, these are individual white flowers. Do you think this plant is photosynthetic? What do y'all think? Just looking at it, do you think it's photosynthetic? The answer is no. Um, so it is not photosynthetic because it's not green. There's no chlorophyll that we see here. And I just told you that it's parasitic. So what it's doing is it's getting its nutrients. I don't know why I said information. It's getting its nutrients usually from the roots that it's tapped into. Okay, this is a native plant in the eastern United States, so it is possible to come across in Mississippi if you're looking at the right time when it's reproducing like this and it pops up out of the ground. Okay, bryophytes. So first group of land plants, terrestrial plants, they are probably different than what you usually think of when you think of as a plant. This includes three groups: the mosses, the liverworts, and the hornworts. And I've got examples of those pictures from left to right. How many of y'all have seen a bryophyte? And which one have you seen?
I've seen mosses.
You've seen mosses? Okay. Those are pretty common. Yeah. So, oh, Elena, you said you've seen them all? You've seen all three? Awesome. That's cool. I love liverworts. These are fantastic. I don't know why. I just think they're so cool looking. Um, they don't get very big. None of these get very big, so they can be easily skipped over. So let's introduce ourselves to bryophytes. There are somewhere around 23, it's probably more than this now, 23,000 species of bryophytes that have been described. This includes mosses, liverworts, hornworts. They range in habitat from sea level all the way up to 5,500 feet or more. So basically any above sea level location potentially could have a bryophyte. The bryophytes of all of their divisions have mycorrhizal fungi associations with their rhizoids. They don't have roots, but they have these things called rhizoids to help them stay in place. And they interact with what's called a mycorrhizal fungus that allows the plant to associate together, kind of a symbiosis, where the mycorrhizal fungus will bring nutrients and water that's further away from the plant to the plant, and the plant gives this fungus photosynthetic product. In a few instances, we see something beyond symbiosis. We see some of the fungi seem to be a little bit parasitic. So not beneficial to both, more beneficial to the fungus. So you'll see something about peat mosses from time to time. They're pretty ecologically important in bogs. There are luminous mosses, but I will tell you, these mosses, from what I can understand, they don't actually light up. They don't. What happens is they live maybe in caves or really dark, damp kind of places, and they basically reflect or gather the littlest bit of light that makes it to them. So they're able to kind of concentrate the light, and it makes them look like they're glowing. But they don't actually produce light, from what I understand. Now, bryophytes are not a natural grouping. They do not include the rest of the tracheophytes. For this to be a completely natural monophyletic grouping, it would need to include tracheophytes, which would be all of kingdom Plantae. So basically, when we talk about liverworts, hornworts, and moss, they are separate lineages on this line that we lump together for ease, to put them just, they're more alike than they're different in some ways. Okay. So we have the division hepatocophyta. Hepatico is liver. Phyta we know is plant. And basically liverwort. Does anybody know what wart, but W-O-R-T stands for, not W-A-R-T? Not like a wart on your finger. Does anybody know what wart means in English? It's Old English, or at least Middle English. Anybody know? Maybe you've heard of St. John's wort. It's not because St. John has a wart, or at least not the wart you're thinking. Any ideas? It actually means plant. Yeah, you're right, Melina. So liverwort is a liver-shaped plant. Hepatocophyta is a liver-shaped plant. So it means the same thing. We think that this one's the most ancestral of all the groups. We then have Anthocerophyta. Just on the off chance that y'all caught the pattern, what do you think Anthocero means? These are the hornworts. How'd you figure that out, Elena? I'm just joking. You found the pattern. Anthocero actually does mean horn, so horn plant, hornwort. Okay. Then we've got the Bryophyta, which is the mosses. I will kind of go back and forth using Bryophyta and mosses because a lot of times I get kind of confused when I'm talking to y'all. You don't know if I'm talking about the mosses or the bryophytes in general. So there's the bryophytes and the Bryophyta. And of course, the bryophyte group is named after the mosses, but they include two other things, the Hepatocophyta and Anthocerophyta. I probably will use the terms liverwort, hornwort, and mosses more often, but on some occasions I will say Bryophyta usually, not the other two here. Okay, so this is where we get our features that we want to pay attention to, just like we got the features of the different algae and the features of general land plants. So in the bryophytes, they have alternation of generations. Not surprising because we've talked about that as true for all plants. What is different than all the other land plants that we're going to be talking about later is that this one has the gametophyte dominant. We've talked about what this means. And if you don't understand, you need to go back and review that because you need to recognize this idea here. So gametophyte dominant, can someone tell me what ploidy level the gametophyte is? Is it 1N or 2N?
One in.
It's 1N. Good job. Gametophytes make gametes, of course. So the plant that we see, when we see like a moss, is a single, it's like all the cells that we see that are gametophytic are haploid. They have one, of every type of chromosome. There is a sporophyte, but it is attached to and dependent upon for nutrition on the female gametophyte. Okay, so it's going to be smaller and attached to it. Some mosses have conducting cells like hydroids and leptoids, but none of them have true vascular tissues such as xylem and phloem. So hydroids, what do you think hydroids move? What do they transport? Water. Okay, so they kind of match up with xylem. Leptoids are those that transport photosynthetic product from the leafy-like areas. So they're more like phloem. They have an epidermis and a cuticle, which is great because we don't see that in the algae. Okay, why would you need an epidermis and a cuticle to be on the outside of your plant? What do you think the purpose is for? We've covered this stuff in the transition to land plants. Exactly, Kyra. It's all about water. It helps to keep in water and helps to reduce water loss. Why does that matter? Well, they're not living in the water anymore. They're living on land. They have air pores for uncontrolled gas exchange. We don't yet see many plants in this group with guard cells and stomata that allow for controlled opening and closing of stomata. Right now, they're just open all the time, which means they have to be really careful of their water loss. They all have a multicellular sterile jacketed gammatangium or multi-gammatangia. So there is a protected area for those places that make gametes, unlike what we see with the algae. So we call the gammatangium for that's male the antheridium, and for the female it's the archegonium. Plural, antheridia, or archegonia. If it's singular, it's antheridium, archegonium. Now, in this case, these plants need environmental water. All plants need environmental water, but this one needs it for reproduction. So to be able to get your sperm to the egg, you've got to take it there by water. So guess what? Do you think flagella are going to be useful in this group? Yep. Okay, that's how the sperm is actually able to move itself to the egg in that water. They're all embryophytic, so they have the gametes coming together within the archegonium, and for at least a while, some development, and most a long while, development of that new sporophyte generation within the parent plant, the gametophyte plant. They do not have specialized mechanical support cells. Okay, so unlike what we see in the xylem that is able to support itself, we don't see that. They're fairly soft plants. They are not rigid. They also cannot be very tall because they don't have that ability to move water very far or support themselves with these mechanical support cells. The gametophytes are attached to the substrate. So in this case, a rock, a tree, whatever the case may be, by rhizoids. These are not roots. Do not call them roots. Roots are vascularized, and these rhizoids are not vascularized. Okay, they cannot transport things over long distances through their roots. There are no roots. They just have these cells that allow them to attach to whatever substrate they are near. Okay, you've probably seen moss growing on a rock, or on the soil, or on a tree. Probably if you stood long enough, you could probably get moss to grow on you. I don't suggest it. That's kind of, extreme. But you don't have to have even necessarily to be attached to something that has water. You just have to have water appear from time to time in your habitat. The gametophytes can be bisexual or unisexual. For those that have bisexual gametophytes, we see that a potential for inbreeding. Those that are unisexual, you can't breed with yourself if you only have one type of gamete that you produce. But you then get gene flow. So bisexual gametophytes, where you have a gametophyte that produces both antheridia and archegonia to do male and female gametes, then you could potentially breed with yourself, and that would be a form of inbreeding. For unisexual organisms like these that are like megagametophytes and microgametophytes, where you've got female gametophytes and separate plants that are microgametophytes, the male ones, you have to cross between two different plants so you get gene flow. Okay. So this is where my drawing skills are going to be problematic. We're going to try this. I'd much rather do this on a chalkboard, but here's where we're gonna do it. Okay, so grab your hands, something to write on. You're gonna want to reproduce this. So let me switch to the whiteboard. Can you all see the whiteboard now? Great. Okay, so these are the this is the bryophyte life cycle. We're going to start with, let's start with the bryophyte. Let's do it here. Okay, so bryophyte, I'm going to represent it kind of like, this is terrible, terrible, terrible drawing, kind of a moss. Okay. This moss is the gametophyte. Okay, this is the adult gametophyte. Can anybody tell me what ploidy level the gametophyte is?
1N.
1N, perfect. Okay. I am going to draw another one over here. Also 1N. Okay, let's make a female on this side. We're not female necessarily, but it's gonna be an archegonium on one side. So let's put some, I'm just gonna make these little circles to represent what's called the archegonium. So archegonia here. On the top portion, I want to use this triangle to represent the antheridia. Okay. Now, these are gametophytes. What do gametophytes make based on their name? Gametes. Okay, so let's make gametes. So what should the archegonium make? It's going to make an egg, okay, which is also 1N. And that means that we went through a process called mitosis because we're going 1N to 1N. Even though we think in humans about doing meiosis to make eggs or sperm, but that's not the case here because we're staying 1N to 1N. For the male side, we're going to have sperm, also 1N. Also, we went through the process of mitosis. Okay. When these come together, we are going to have the process of syngamy, which is going to create a zygote, which is, when you put 1N and 1N, you get 2N. Okay. This will go through the process to produce an embryo. I'm not going to try to draw an embryo. Embryo is going to still be 2N. Okay. So if we went from 2N zygote to 2N embryo, what process do we go through here? Syngamy? mitosis, meiosis, meiosis, whichever you want to call it. So we went from zygote to embryo. Which one of the above? It's not meiosis. We stayed the same ploidy level, 2N to 2N. So therefore it has to be mitosis. So when you stay the same level, 2N to 2N, it's mitosis. If you go up, in this case, 1N to 1N together, syngamy, it's this process of syngamy. So 1N, 1N gives us 2N. We went up in ploidy level, it's syngamy. If you stay the same, it's mitosis. If you go down, it's meiosis. Okay, so our embryo continues to grow. It will eventually become a I'm going to have to do this a little bit strangely, but I'm going to take it all the way over here because this is where the female's at. Remember, the water will have splashed our sperm from here into some of these archegonia. So from one of these archegonia, we are going to have our sporophyte. So this now is our sporophyte. So the embryo developed through mitosis again. Okay, now let's label some of these pieces. So where it connects down here, it's called the foot. This kind of long piece here is called the seta. And this little top of the, kind of looks like a firework to me, the firework part is called the capsule. We'll look at the capsule a little more in depth later. So those are kind of the three main parts of your sporophyte. Now, inside of this capsule, because this is a sporophyte, what do you think is going to get made inside that capsule? When this top blows off, what's going to come out? So the name is sporophyte, so you must make what is it? Someone tell me. It's going to be spores. Yeah, exactly. Okay, so these spores will, inside here, they're developed from something called the spore mother cell or sporocyte, which is 2N, that will produce our spores. Okay, we're going to be general here because sometimes it's going to be a microspore, sometimes it will be a megaspore if it's male or female, but most of the cases it's just going to be, some of these early plants at least, it's just going to be a spore. And a spore is 1N. So what process do we go to go from 2N to 1N? Meiosis. Great. And I'm going to just put this here. Okay. So we've created spores, and spores develop into gametophytes. And we start the process all over again. I should also add here, hold on, device R1N. Do you have any questions? No. Do you think you could draw this? Okay. Remember that we practiced with a basic idea of alternation of generations the other day. And this is just the alternation of generations repeated again, but just within the context of the bryophytes specifically. So we do want to keep that in mind. Okay, let's go back. Cool, just let me click, great. Huh, interesting. I see how that works. Okay, gotcha. Okay, let's go into the details and not just the general bryophyte understanding. So in the liverworts, the gametophyte is dorsoventrally flattened with rhizoids on ventral surface. And you're like, what on earth did I just say? Dorsoventrally. So you may be familiar with the ventral surface and the dorsal surface. If I were to put a dorsal fin on you, where would that be on your body?
Would it be by ribs?
So are you talking like right here on the side?
Yes.
Okay, that's not where it would be, because think about a dolphin. I mean, they don't swim sideways.
Oh, yeah, on the back.
Yeah, it's going to be on your okay. I don't really have anything that looks like a dorsal fin, but I don't know if you see my book or not, but that will be my dorsal fin. So this is my dorsal surface. The ventral surface is going to be here. Okay, so dorsoventrally flattened would mean that I would be laying down on my belly. Okay, so I got to ask, how odd is it that I just did that on video? But did it help? Did it make sense? It got me worried. I'm not hearing anything. You're still on the whiteboard. Okay, let me see if I can. Let me stop the share for a second and try to figure out what happened, why the whiteboard stayed. I thought I switched it back. Okay, are you on the whiteboard now, Elena? Okay, so dorsoventrally flattened, real quick. Dorsal surface is my back, my ventral surface is here. If I'm dorsoventrally flattened, I'm going to be laying on my belly. Okay. There's going to be rhizoids on that bottom. So are rhizoids roots? Nope, they're not. Okay. So many of our gametophytes are going to be like this. They're going to be laying down. Rhizoids on the bottom. This is their dorsal surface. There are two kind of shapes that you'll see. The phallos shape, which basically doesn't look like it's a stem or a leaf like anything. It's not really any organized shape of any sort. It's basically just a mass of parenchyma cells, but they do have a dichotomous branching where you'll see, that's not what I wanted. I want the pen. There it goes. Okay. It does this. Okay. So they keep dichotomously branching, always branching into two. So it's always this shape. And then they have another kind called the leafy liverworts that look like they kind of have leaf-like structures. Now, they cannot be leaves because they're not vascular. So can we use the word leaf in the bryophytes? No. What we say is a phallidium or multiple phallidia. And hopefully this will make sense. Chlorophyll, where do you find chlorophyll? You find it in leaves, regular plants. So a phallidium is basically saying it's a leaf-like structure. And stem-like structures are called caulidia. Okay, called is stem. Maybe you've eaten cauliflower. Okay, so that's got a reference to stem in the name. We have air pores, very common, with epidermis and cuticle, not surprising based on the group that it's in. There are no mechanical support cells, so it doesn't get very tall. It's pretty short. Asexually reproduction is really, really common. We're going to see this under the microscope in lab. Fragmentation is very common. They just break apart because they have that dichotomous branching. They'll just pull apart, and a new one can make a whole new plant. They have gemmy. By the way, these are called gemmy cups, Gemmae. These are asexual spores, and you'll see them, like here's one here on the right. So this is a gemmy cup. And they have these little asexual reproduction taking place. And when water splashes on it, they'll splash out and disperse, and then they'll grow whole new plants. Now, if it's asexual, we're talking about making basically little clones of this plant really commonly. Okay, I just realized it is 8:51. I'm talking too slow. I have to come back to this later. Sorry, guys, I didn't realize what time it was. Okay, I got to stop there. I'm going to let y'all go. I will not see you Monday. I will see you Wednesday. Y'all have a great fall break. And if you have any questions, I'll be here for a little bit. So otherwise, see y'all later. I'll get the videos put up as soon as I can.